We are on with proteins today. So this is the A-level biology, the third topic in unit 1. So with proteins, what do we need to know? We need to know its primary structure. Okay, so primary structure. So you need to be able to draw this in the exam. All proteins have a carbon in the middle. They have an R group, that's a variable group. There are 20 different R groups, making 20 different amino acids. Then there's a carboxyl group. This is a carboxylic acid, and the NH2. This is an amino acid. It is called an amino acid because the amine group is there and the acidic group, the carboxylic acid, is here. Like I said, there are 20 variable R joints, and these 20 variable R regions make 20 different amino acids. In the entire universe, there are 20 different amino acids. This is a protein's primary structure. So, to answer the question, what is a protein's primary structure? You literally just need to say its sequence of amino acids. Now, we have amino acids, like I've mentioned, and I'm going to draw out another amino acid. So the variable R group, the H, the COOH, and the NH2. Now, I said a primary structure is a sequence of amino acids. These amino acids, like we've talked about in carbohydrates, need to bond together, don't they? So when they bond together, when bonds happen, it's called a condensation reaction, as you already know. So condensation means to make water. So look, H2O is going to be made. So we need to remove 2 hydrogens, 1, 2, and we've moved the hydrogen there, and then 1 of the oxygens. Okay, so now if we redraw this, we're going to have an oxygen here, and then the carbon and the nitrogen join, and I'm going to rebond the hydrogen to show it properly. And here, this all is fine. The carbon is bonded 1, 2, 3, 4 times. The nitrogen is bonded 1, 2, 3 times. All good. The bonding is perfect. We've lost water. We've formed a bond here now in this condensation reaction. This is called a peptide bond. Peptide bonds form between amino acids. This is what makes the primary structure of an amino acid. Next then, we're going to go on to the secondary structure. So the definition of the secondary structure is going to be 2 types. One type is called alpha helices or alpha helixes. Okay, so alpha helices is how the primary structure forms these helical shapes like this. So a helix is kind of like a wavy, bendy shape. Okay, so the protein sheets can fold into alpha helices. We need to know the type of bond in alpha helices, the type of bond that exists between molecules, specifically the NH molecules, is going to be hydrogen bonds. Now, these aren't covalent bonds like you've been taught about in chemistry. These are intermolecular forces. So, individually, they are quite weak, but together they have this Velcro-like effect, and it takes many of these bonds and a lot of energy to break the bonds. So they actually are quite strong. So these exist between the N and the H, the N of one amino acid and the H of another amino acid. So the NHs of adjacent amino acids. That's what exists and allows hydrogen bonds to form. The second thing then, or the second secondary structure an amino acid can take, or a protein can take, because we're on with proteins now, is going to be beta pleated sheets. So these are helices, and the next one is going to be like wavy sheets like this. So these are still 2D per se, and once again, you're still going to have H bonds, hydrogen bonds forming, okay? And this is all going to give rise to our tertiary and quaternary structures. Secondary structures is quite simple. It's just the formation of hydrogen bonds in the NH groups, okay? Next then, we are going to have our tertiary structures. This is the most detailed one, and you need to know about this the most in the exam. So, with our tertiary structures, this is a specific 3D folding of a protein. Okay, now I should mention we have amino acids in a chain. I've said one amino acid is a peptide. I said one amino acid is the functional single group of a protein. Many amino acids makes a poly. Poly means many, and peptide. Many peptide bonds. Polypeptides are proteins. Okay. So here we're making polypeptides, and we made polypeptides in the secondary structure as well. So we have the specific 3D folding of a protein in the tertiary structure, and we need to know the type of bonds or intermolecular forces as well that exist between the amino acids in the tertiary structure of a protein. So the types of bonds / forces. And this is dead straightforward, okay? So the first one is hydrogen bonds still. That's intermolecular force. We have one called disulfide bridges. So this exists between some of the R groups we've talked about has sulfur in it, so we can form sulfur bonds between two different R groups, provided they have sulfur in it. We also have another type of intermolecular force called van der Waals forces. Okay, van der Waals forces. We also then have ionic forces of attraction. So some of the R groups allow, because they're variable, remember, there are 20 different R groups, we allow ionic forces of attraction, all keeping the tertiary structure strong. And then there are also hydrophilic, which means water-loving, and hydrophobic, water-hating interactions. Okay, so the 5 things that can arise in the tertiary structure. All 5 of these then will give rise to the tertiary structure. I'm going to write 3 structure. So what actually is a tertiary structure? It's a specific 3D folding of the protein. Now, the tertiary structure matters so much because proteins entirely depend on their shape. For an example, an enzyme has to have a complementary active site to catalyze its substrate. So let's draw this enzyme like Pac-Man, and let's draw a substrate like this. This enzyme's Pac-Man shape has come from its tertiary structure, which is why the tertiary structure, the specific 3D folding, caused by H bonds, disulfide bridges, van der Waals forces, ionic forces of interaction and attraction, and hydrophobic and hydrophilic attractions, matters so much because it allows for a specific 3D folding which can catalyze the substrate. Let's say the 3D folding changed and made it into this. This is no longer complementary. Active site enzyme substrate complex can't form, and the reaction will not be catalyzed, okay? That's why it matters so much. That's a tertiary structure. Finally, we're going to move on then to the quaternary structure. So, not all proteins are quaternary. Quaternary proteins, some would say, are highly specialized. So the feature of a quaternary protein is 2 or more polypeptide chains joined together by bonds. Okay, 2 or more polypeptide chains joined together by bonds. That is what makes a quaternary protein. Now, quaternary proteins are typically associated with something called conjugate groups, or more commonly known as prosthetic groups. So you think about prosthetic limb. Prosthetic limbs is something that's like, it's like a fake limb. It's not a real limb. This is the exact same here. It's something that is a non-protein derivative associated with the protein structure. What do I mean by that? Let's go off our definition. 2 or more polypeptide chains joined together by bonds. I'm going to draw this now. 1 polypeptide chain, 2, 3, 4. This is a molecule known as hemoglobin. Okay, so hemoglobin has 4 polypeptide chains joined together by bonds, so it checks off this definition. And look, also they sometimes have prosthetic groups or conjugate groups. As the example of hemoglobin, the conjugate group or prosthetic group is the heme molecule or the iron bit. So this is a non-protein associated group. It is an ion, which isn't protein associated, because remember, go back to our primary structure, this isn't an amino acid. So this is highly adapted to its function. The iron allows oxygen to bind in the example of hemoglobin. So then hemoglobin in red blood cells allows oxygen to be carried around the body and delivered to places that need oxygen the most. So that's an example of a quaternary protein. Hemoglobin is one. We also have chlorophyll in the chloroplast of a plant. That is bound to magnesium, which allows for photoionization. More on that in year 2. Okay, so this is what a conjugate group is. Now, a really, really common exam question they're going to ask you is, how would a change in primary structure change like tertiary structure? And it's dead simple, this question. So you always, to answer these questions, you want to start it off with change in primary structure leads to change in amino acid sequence. Therefore, change in polypeptide chain. Therefore, different like intermolecular forces / hydrogen bonds / like dipole forces / ionic bonds, etc. We've talked about them in the tertiary, quaternary. Therefore, different specific 3D folding in tertiary. Therefore, different shape. And remember, proteins depend entirely on their shapes. I give you an example of an enzyme. Proteins are also hormones. Hormones depend entirely on their shape. Everything you need to know about proteins, it depends entirely on its shape, okay? So that's kind of how you talk about a change in a primary sequence can lead to a complete functional change in a protein itself, okay? And this is all to do with the idea of shape and changing its primary structure and the bonds and forces that arise from that change itself as well. What we need to know now then is a test for proteins. So this is called the Buret's test. And basically what you do is you have a sample in a test tube. Let's label this X and let's pretend X has protein in it. Okay, so it's got protein. We add Buret's reagents to this, and the observation is it goes from a purple lilac color. So it turns purple or lilac, okay? And this indicates a positive test. This comes up as a 2 marker quite often, so it's worth knowing the Buret's test, okay?